So we next want to look at polymerase chain reaction, or PCR. And so in PCR, we can have a very tiny amount of a cellular sample or a source of DNA and amplify it very, very easily and quickly. So we can make really millions upon millions of copies of a segment of DNA within about 2 to 3 hours. And so we use PCR then to be able to amplify DNA from a genomic library. We use it in sequencing DNA, which we've seen with our Sanger sequencing or pyrosequencing. We can use it for forensic analysis when only small amounts of DNA are available. For instance, at crime scenes, or if we just have a little bit of hair or sperm or blood, we can use just a small amount of cells to identify someone then. As well, we've identified some mummies. And I think the most recent big name that we've found someone by their DNA and PCR is King Richard III of England. He was a king who was killed in 1485, and they never found his remains until just a couple years ago. His remains were found in, I believe, a park when they were doing some type of archaeological dig, and they identified him via PCR and based that DNA sequencing then on the DNA of his descendants or distant relatives. We can also use it for DNA fingerprinting. That helps us to ID people based on sequences called SNPs, and we also can use it in disease diagnosis. And so for those of you who have heard of like BRCA1, BRCA2 gene analysis, when breast cancer patients come into the clinic and we test them for the BRCA genes, we use PCR. So to do a PCR, we do a reaction, and then we put this reaction at different temperatures. And so to begin with, in our tube we have different components. I call it the PCR ingredient list, and we want to understand what each of these are so that we understand how the reaction is carried out. So the template DNA is the actual sample. So this DNA is isolated from our cells, and we don't need a lot of it, so we only need a tiny bit. The enzyme that carries out our reaction is DNA polymerase, and in PCR reactions we use an enzyme called Taq. Taq is expressed in an extremophile, a bacterium that lives at very, very high temperatures. So this Taq polymerase, since it's an enzyme, and it's in this bacteria that live at high temperatures, the enzyme doesn't denature. That means it doesn't unfold at high temperatures, and that is key for PCR reactions occurring properly. We also have to add in our DNTPs, so this is our A, T, G, and C, so that we have these reactants available so that polymerase can add them to a growing nucleic acid chain. And we have primers. So primers are small fragments of nucleotides that match or areas before and after the gene of interest. So we have to know something of the sequence before we do a PCR reaction. And then we have buffer, so it's the fluid that our reaction is going to be carried out in. And so if we look, PCR is a cycle, so it's going to occur multiple times, and it will include increasing and decreasing our temperature. And so here you can see is our original piece of DNA. This DNA is going to be heated, and as it's heated, that is going to break the bonds between our two original strands, and we will then cool the reaction. So we drop the temperature quickly. And as we do that, that will allow these primers to anneal. And so you can see the primers are annealing in the right orientations. Remember, DNA replication occurs 5' to 3'. And so this 3' nucleotide, the 3' carbon is open, that OH group. That will allow our polymerase enzyme to start adding subsequent nucleotides to it. So then the dNTPs that are in our reaction, they will be incorporated into and matched to their base pairs along our template DNA. So our enzyme is going to run and base pair these nucleotides all the way down the length of the DNA. Then what we do is we heat our reaction again to a very high temperature, and all of these strands that we've just made, here you can see is that primer again, and here's the original template, they are going to separate, and the reaction will carry out again. I think a video is worth 1,000 words, and so we'll check out a video.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.